Dr. Sinha was one of the early recruits for Dr. Sahel when Dr. Sahel arrived here in Pittsburgh in 2016. And Dr. Sinha came from Johns Hopkins University with a very large team of scientists with him and doing some very important work, which we're going to talk about here shortly. But Dr. Sinha, welcome and thank you for sitting with me today.
Thank you very, very much for having me. Good afternoon.
Well, you know, listen, you know, I always enjoy getting to see you, and every day that I do, I always learn something, and there's a lot going on. And you know, all of our faculty, and you're no exception to this, have come from very diverse backgrounds and places around the world and have done a lot of education and training in some of the most prestigious universities around the world. And again, you're no exception to that rule. But, you know, I love to hear a little bit about people's journeys, just a bit. And, you know, along the way, as you were going through your education and training, you made a decision that you're going to focus on research and as well kind of focus on how you can impact people the most through your research. Can you tell us a little bit about that part of your journey?
Yeah, sure. Thank you for the question. So, you know, I grew up in a research institute. So my dad was the division director of a big institute in India, the Council for Scientific and Industrial Research. It is like NIH in this country, but he was a physical scientist. So, you know, but I grew up with scientists, and then I like to read. And so I read a lot of lives and, you know, biographies. And what really attracted me in a very early stage of my life was the work and life of Madame Curie, Marie Curie, Barbara McClintock. And so I was actually poised. There was nothing else that I tried and came back. So I wanted to become a scientist. So once I graduated from India, I came to Medical University of South Carolina in Charleston, South Carolina, to work with one of the very amazing scientists, Dr. Lisa Matsuda, where actually I worked on the cannabinoid receptors in the brain. And she was the first person, so the lab was the first lab to show that there is a receptor in the brain. So before that, it was not there. So after that, you know, unfortunately, she left science. So I had to move. And I landed up at National Institute of Health, working with Dr. Joram Piatigorsky at the National Eye Institute, right? Mm-hmm. So I was there a postdoc for about 3 years, and I actually married very young. And my wife was very particular that I don't become a lifelong postdoc. So I went to and worked for a company for a few years, you know, and I actually, after 4.5 years, a company that is now was taken by Pfizer, I did not like it very much. But I have been lucky all through my life, you know, meeting people who have really helped me out. I'm here today only because of luck, you know, nothing else, because many people down the road has helped me so much. So then I luckily, because from academia to biotech companies or pharmaceutical companies is very common. But the opposite direction does not happen because it is difficult. But I was lucky enough to get a position at Johns Hopkins Medical School, where, you know, the then chairman of the school, Dr. Mort Goldberg, was really helping me out. So I had a good time. And then now I am here in Pittsburgh because of Professor Shoel. So that's my journey to Pittsburgh.
Well, and you know, I certainly remember when you were recruited, it was a very, very exciting moment for all of us here to have a strong scientific focus on macular degeneration and causes of vision loss in the retina, and it's really what your lab is focused on. I'm gonna, if you could tell us a little bit about just the you know, and I'm gonna say the 30,000 foot view of your research, meaning in a way that you know, our listeners who may not have the scientific education would understand, because it's compelling for sure that what you're doing. But if you could just share with us sort of how what your target is and what the possibilities are in terms of what you're looking to do with research to help patients with their vision loss.
Yeah. So, the major focus of the lab, as you mentioned, is in age-related macular degeneration. So we work on the back of the eye. And in the back of the eye, as you all know, that there is these photoreceptor cells that get the light. But then there's the one single layer of cells called the retinal pigmented epithelial cells. They absorb the light. There are different functions. And in the last 2 years, we have identified that these cells, so in age-related macular degeneration in AMD, there are two forms. There is a dry form and a wet form. So there is treatment for the dry form, right? You get anti-VEGF from Ayala or any other companies. And so there is some sort of treatment. It's not 100%, but there is something. But, for dry AMD, unfortunately, there is no treatment to date. And again, unfortunately, 90% of the AMD patients suffer from dry AMD, not wet AMD. Okay, so we are focusing on these cells because these are the cells that, like macrophages that you have heard in the body, that engulf bacteria when you get sick and you help, in the COVID-19 period, you are listening of infection, inflammation, and everything. So in the eye, these are the cells which does the major work of it. And they actually every day engulf 40,000 in the rats, these outer segments, to have the visual cycle going on, to make the eye healthy and everything. So one of the things that we are very interested are two things. One is called phagocytosis, self-eating, and then another is autophagy that happens, in the cell completely. It's like clearance, right? And both these clearances in any cell happens through an organelle called lysosomes. So lysosomes were known as waste disposal organelles, right? So it's like in your body, in your house. If you have your waste building up, then definitely what will happen is that it will stink, it will smell, it will, so this we figured out that we do not know, and it is not the only reason, but we figured out that one of the main reasons why these drusens form may be because the lysosome does not function correctly, okay? And when they don't function correctly, there is deposits that are, in the eye, in the back of the eye. That's what is called drusen. So clinically, when you go to a ophthalmologist, they will say, oh, we detected drusen. So the idea is that somehow, so all our targets that we are building now with different companies and at NIH that we are with our grants is to rejuvenate the function of the lysosome. So we are trying to clean the waste and trying to see. So the idea is that it will be amazing if you can cure the disease, but at least what we are thinking that we can delay the disease progression. So this disease, as you know, is a real aging disease. It's actually around 60, 65, 30% of the population gets it when they are 75. So our idea is that if we delay the progression and to 10, 15 years by rejuvenating the lysosomes, I think that would be enough right now for a whole population who suffers so much and can go blind, and to have something to look forward to. They can see and have better vision with this thing. So that is what we are trying to do with different targets. The other thing also we do is also diabetic retinopathy. We have recently started doing in the lab that we work on a type of cells called glial cells called astrocytes. These are the cells which are also affected in brain tumor. The maximum brain tumor are affected by cells. So we are the first lab that we are showing that these cells are very important in diabetic retinopathy. So we will be trying to target these cells in different ways. So these are the two main disease targeted research in the laboratory.
Well, it's exciting, and I know that recently you've been able to actually secure a relationship to help maybe get some of these targets that you're working on and these therapies and treatments that you're working on to patients a little sooner. What is what would be your most optimistic prediction in terms of how soon we may be able to see some of these therapies in clinical trial?
So, as you know, that we recently, University of Pittsburgh recently signed a contract with Astellas, and we are extremely happy because of that. The reason being, it is very difficult from a lab like mine or any labs per se to go in to take a drug through real in the market, right? It's like it costs about $1 billion easily at the minimum. So we are very fortunate again going to Astellas, and we were very careful. We had many other options also to take because many people were interested in our target, but we wanted to have a company or a pharmaceutical industry who would be serious about it, who would be serious about ophthalmology, who would not just tank it after one or some years and say, okay, this is not our interest anymore. Thank you very much. Okay, so we didn't want. So Astellas is actually very serious about, not only serious, it is now their number one target of whole of Astellas, like not only ophthalmology, the whole thing. So that means, and so that gives us the impetus that it will actually drive much. So I'm thinking, we are slowed down a bit because of the pandemic situation, because we cannot travel, we cannot do as much. Of course, Zoom is good and everything, but as you know, research is a benchwork thing. Not everything can be done through Zoom, and it's better always to discuss face-to-face than anything else. But the way we are progressing, I'm very hopeful, and they are also wanting to do it, that maybe we will go sometimes next year, this time around fall, we may be able to go into phase 1 clinical trial.
That would be amazing.
Sorry?
Yeah, that's amazing. And that would certainly, it just kind of is a testament to really, it is resources. I mean, we can, it's possible to send people to the moon, but without resources, nothing happens.
So Astellas is a very big deal for us, and I think that it is very interesting. They are very collaborative. They listen to us. It's not like they are dictating us on anything. They know. Now recently they have purchased a huge gene therapy company. So part of that gene therapy is coming to us for our product. So it will be that where it will be, going into. So we are very excited, and the up to now, touchwood, everything looks very promising. But as I told you that before many times, that you never know. There are obstacles in drug development. It is not like, oh yeah, everything is.
I know, right, absolutely.
But we are excited, and then with Hoffmann La Roche in Switzerland, we are also trying, because they have some drugs in the market from their immune regulation for inflammation and everything, that we have actually found to be working in AMD also. So we are trying to go into that. So maybe in a year and a half or something, maybe they will also take those, and that is easy. Both the things will be a little bit easier if it works, because since the Astellas drug, for dry AMD, there is no therapy right now. So if going through FDA might not be difficult if you can show all the data and the particularly, everything that are in stage that they want. And for Roche, if it works, really what we see, and we have amazing animal models, that's what we have. So if it works in those, then because it is already marketed in inflammation and the immune regulation, this would be much faster also to go to the market. So for both the things, we are very hopeful, and we are human beings. So survive with hope. So, hopefully it
Ye-
will happen.
That's all we can always provide is
Yeah.
hope, but that's giving us a lot of hope. So thank you.
Mm.
Now, you've done many things already in your career. You're very accomplished, and you've certainly collaborated with people all over the world. If you were just to tell us one thing, and just sum up what you feel the most proud of in terms of things that you've accomplished, what would that one thing be?
I think I have not accomplished anything yet. One thing would be if any of these drugs goes through phase 3 trial and comes to the market and helps the patient population. Because I was telling you before this that, one of my very good friends is Jim Handa, who is the division director of retina at Johns Hopkins, who is a very fine clinician, scientist, and AMD. So at a few times I have been to his clinics and has seen AMD patients and seen the reactions. I was just telling you after the Astellas things came out, like I saw how much every email that I'm getting, that how much people are just waiting. It's amazing that it is such a difficult situation that, we talk about numbers, we talk about everything, but it is very, very difficult to understand what is thing. Again, I think all this is happening because somehow, scientists don't believe in luck, but I think I'm very lucky. Every step I had amazing people, and this department is amazing under two really very strong leaders. Of course, Professor Shohel doesn't need any introduction or anything. Everybody in science knows him. But, Jeff Gross also. Jeff is an amazing research director who himself is because if you look at ophthalmology departments and research directors, he has qualities that is very different. He's young, he's himself an extremely great scientist, right? He understands, and it is very important because we have one of the youngest group of scientists maybe in ophthalmology in the whole world right now. And to do that, you need, and he's a bench scientist, right? So for a young guy to talk to, for me, it is okay. I can tell him, and he's always supportive of me. But for a young guy, scientist to adapt and to know, he is perfect. So I think that it matters. So I'm, again, very hopeful that in a 5 to 10 years time, this will be one of the amazing ophthalmology departments in the world.
Well, I think, that actually is answers even the other questions I was going to ask you, is essentially where you see us going. And I think that's actually exactly the answer that I think we're all hopeful for as well. I think, what Dr. Sal has already brought, and you are a very key component to that, is is a very good mix of individuals who are working very well together. And I'm always so pleased to have a chance to catch up with you and hear some of the work that you're doing, and what an exciting time. So thank you for all you're doing. Thank you for your contributions to science, your contributions to the department, your wonderful, engaging personality that's always very, very enjoyable to spend time with. And thank you for spending time with me today.
And thank you very much, Lonnie. Thank you for the support from INER. Again, as I said, everything happened today because of people like you and Professor Shoel and Professor Gross and my lab members and the whole department. It's a different, we have a retina meeting. If you have time, just come one day. You will see how engaging we are, and everybody is looking for that cure.
I'm going to do that. I'm going to do that. Well, thank you so much, and we'll wish you a good day.
You too. Thank you.